Okay. So, hello, my name is Jeekin, and for those of you that might not know me, I'm a postdoc for Oscar, and I did my PhD with Dave, and I've kind of been here since the first round of the Rosette grant. So the first round started in 2014, as you can see on the bottom. I joined in 2015 and have been here ever since. So I'm going to talk about fungal pathogens in the diploid and autotetraploid mapping populations. However, since I have been here since the beginning, I have one slide on the RRD resistance. So it was a long project. I mean, we were making crosses in the dark. We didn't know what parents were resistant or were susceptible. When we first got the grant in 2014, that's when we put out the cultivar trials to see what were resistant. About halfway through the grant, we were like, okay, maybe we have some populations that we've been crossing in the dark that actually will segregate for resistance. So it was a long process. The populations that were eventually placed out here, where we just were, they were ripped out and replaced with the new things that we saw today. Those were crossed in 2016. They were propagated in 2017. They were planted in 2018. They were phenotyped for 3 years. And then Sarah did all the PCR. She did all the phenotyping. And then in 2022, finally, we have this string of papers. And Tessa's going to go in more depth about these papers and her paper. But what we generally saw circled in the blue, those were the diploid QTL that we found. Circled in the red were the tetraploid QTL. So the first paper that came out was in the diploid level. A couple months later, we came out with the tetraploids. And then Tessa wrote up a meta-analysis on kind of trying to combine what we saw, because we saw some overlap in linkage group 5, linkage group 6 between the diploids and the tetraploid, and she kind of did that. I'll let her talk about that next. So a little bit of background on roses for those that aren't familiar. We heard earlier that the economic impact is really big. In the U.S., garden roses, the most recent census is close to $170 million value. Important breeding goals for any breeder would be disease resistance, flower productivity, because you want plants that look good in the landscape, and plant architecture. And talking about plant architecture, just look at these two pictures. Dr. Bird on the right is holding some miniatures that we found in the greenhouse out of some, I can't remember if it was mapping population or just general breeding stuff, but we found some miniatures. And that's him in the picture on the left up in Chambersville, standing next to these climbing monsters. So there's great diversity in roses, which leads to the genetics. The great diversity in roses comes from the fact that the cultivated rose and the landscape rose kind of are interspecific crosses that come from 10 different species, and they range from diploid up to decaploid. And because of these interspecific crosses, they're highly heterozygous. The modern garden rose that we would find in Lowe's or Home Depot are diploid, triploid, and tetraploid for the most part. And on the bottom, those are just some root squashes that me and Ellen did really early on when probably it was my first and second semester. We struggled a lot, but we finally got them to work and we could actually count chromosomes. And that was another unknown. We were making crosses and you can't really tell a triploid from a tetraploid just looking at it in the field. So we had to really make sure that we were making crosses that would be fertile, making crosses that would be able to be mapped in mapping populations. A little background on some of the funguses. So black spot is caused by a fungus called Diplocarcin rosea, and it causes black spots, real creative. And these black lesions are feathery on the edges, and it's favored by hot, humid conditions. And severe infections tend to defoliate the plants. What happens is these little lesions start really small. Then as they get bigger, the leaf turns chlorotic, and then the leaves just fall off. And in Texas, it's hot and humid, and it creates a great screening for this disease. And in addition to it being hot and humid, we have overhead irrigation to make it more humid. Black spot has been studied a lot, and all those papers down below. We've heard about vertical resistance. We have RDL 1, 2, 3, 4, and we just heard about 5 and 6. And there's been horizontal resistance that has been described. There has been a meta-analysis done that was done by Lopez et al., who just spoke. So there's been a lot of work done on this pathogen. On the other hand, Cercospora, we haven't seen as much work done in this fungus. Really, there were only 2 works done, one in 2012 and the one in 2020 was a master's thesis out of our lab. And it's characterized by these circular lesions, and they're necrotic in the middle, they're tan, and they can be, they're a lot more circular, and it's not as feathery of a region, of a border. And very little work has been done on this, and literature seems to say that extreme infections can defoliate the plant. However, maybe it's the strain that we have in Texas. I've seen plants covered like 90% with Cercospora, but retaining their leaves, or retaining the majority of their leaves. So the next thing we like to look at in our plots is also defoliation. We've kind of heard about that as we were talking about the app yesterday, how people can rate it for black spot for defoliation. And as a homeowner or as a landscaper, you want to have plants that look like the one on the left that have a lot of flowers completely covering the shrub, and you have leaves. And you don't want one that's all to the right, where you kind of really, they look like twigs coming out of the ground. Kind of like what some of the roses looked like earlier in the field when we went to go look. And defoliation can be caused by biotic causes, mainly black spot, I guess literature says Cercospora, and by abiotic causes. Abiotic causes, mainly heat stress and drought stress. So we aim to try to study those. So on the diploid level, there's been 2 papers, I believe both of these came out this year from Zena, another postdoc in her lab, and she took, in these 2 papers, she took really historical populations that have been developed out of our lab, and they were really big populations that were kind of interconnected, and the next slide kind of shows you what they are. So there's a TX2WOB, it's 11 F1 populations that are interconnected, and Mandy Yan did a lot of that work and established those and did a lot of the first genetic studies using SNPs in our lab. And the second set was the TX2WSE populations. There were 6 F1 populations from Ellen, who many of you remember. And those same populations we're planning to here to look at rose rosette and stuff like that. To kind of give you an idea how messy these are, this is the TX2WOB multi-parental family. So it's a mess. The blue and red, so the red lines are the females and the blue lines are the male parents. So this was Mandy's populations, and these were Ellen's populations. And so what Zena did is she took all this data and she kind of looked at both black spot resistance and Cercospora resistance. So these populations were genotyped using a GBS platform, using RADseq. And for the 2 sets, 2 consensus maps were created. And how those were created were the largest populations in each one of those sets had linkage maps made for them, and then they were combined, making a consensus map. And then from those consensus maps, she used FlexQTL to do the QTL mapping. So black spot and Cercospora incidence, or the same scale that Dr. Byrne talked about earlier this morning, kind of a percentage scale where 0 would be none of the plant was covered by the pathogen, one being about 10%, two about 20%, so on and so forth. And these populations were collected for about 3 years in various places, either in Overton's, in just a lot of different environments. And she combined all of those together. And generally what she found is that the narrow sense heritability was low for black spot and moderate for Cercospora. The broad sense was moderate for black spot and high for Cercospora. And black spot showed a little higher G by E effect than Cercospora. And a low negative correlation was found between the two traits. And it's kind of important just to note that, because we kind of will see the same thing on the tetraploid level. Kind of similar to the stuff that Dr. Burns said this morning. So what Zena found was about 28 and 38 QTLs. So 28 for black spot, 38 for Cercospora, just all over the 7 chromosomes. And the most important ones, though, were on linkage groups 3 and 4 and 6, I believe. But these two are just pictures of 3. The one on the left, let's see if I can get a pointer. The one over here, the ones in red are Cercospora. So these QTL. The ones in blue are black spot. And you see how they're overlapping? That becomes important in a little bit. The same thing in this next population. You have the Cercospora QTLs in red and the black spot ones in blue. So just notice that there's these overlapping QTL there. And remember, I said there was a slight negative correlation earlier. Looking at the other linkage groups, what we have are on linkage group 4, a bunch of Cercospora QTL, linkage group 6, a bunch of Cercospora QTL. And when we did the haplotyping, or when Zena did the haplotyping, what we see is these were for the QTL on linkage group 3, that these haplotypes were the same as these haplotypes. These up here are black spot, and this is Cercospora. So the haplotypes that had lower black spot had higher Cercospora, and for the other population, these that are circled and these that are circled are the same haplotypes. When you had lower black spot, you had higher Cercospora. So earlier on these 2 slides over here, what we see here, these overlapping QTL, these were actually Cercospora susceptibility QTL and black spot resistance QTL. So for both of those, it was that way. On linkage groups 4 and 6, these were just other haplotypes that she found for Cercospora. In this case, the QTL found on 4 and 6 were resistance QTLs. So the conclusions on the diploid side is that major resistance QTL were found, and they co-localized on linkage group 3. And unfortunately, they are co-localizing in different directions, and they have different effects. Cercospora being susceptibility allele, black spot being a resistance allele. And major loci were found on linkage groups 4 and 6. And what she found was that J14.3, one of the breeding lines out of our program, was a source of black spot resistance. And OB Oblush, I think maybe is OB Oblush, I don't know. Violet and M44 were sources of Cercospora resistance. So on to things I'm more familiar with, because this is work that I did. On the auto tetraploid garden side, the garden row side, I'm sure y'all seen this slide before, but these were what were done. These were the populations that were used in the RD studies, and they were crossed in 2016. They were propagated in 2017. We planted them in 2018, and we had the stormy weather by Bright Eyes population of about 200, and Bright Eyes by my girl with 157. It says 3 sites here. I guess I never changed that. They were in Tennessee for Rose Rosette. They were in Somerville and Overton for black spot and Cercospora and defoliation. This material was genotyped using a SNP array, because at that time we weren't really able to do genotyping using GPS and be able to deal with it on the polyploid level, because we have to call dosages for the heterozygote classes. But now we're being able to do that. We're gaining the ability, and we're still trying to iron out things, but it's more of a possibility. We mapped things in Polymap R. I imported that map that was mapped in Polymap R to Map Poly to create the input for QTL Poly, which is where I did the QTL mapping. And I displayed the results using a new package called View Poly that another postdoc in our lab, Chris Taniguchi, made to help us visualize some of this stuff. So the linkage maps that came out of this were pretty dense, around 5,000 markers on both of the maps. And on the tetraploid level, we just had this one paper that talked about black spot cercospora and defoliation all together, and kind of talked about the interplay between these traits. And this is probably the proudest figure that I've ever made for a publication, just because it's pretty. But what you see on the x-axis is the phenotypic scoring from a 0 to a 9. It says 10 there, but that's just the end of the axis. And what you see here are we took the data monthly over the course of 3 years. So for here, this would be the distribution of black spot in 2019 in the 6th month, in the 7th month, so on and so forth. And we just see the year, that would be the yearly blues, or the yearly average of the phenotypic distribution. What you see is that for black spot, this first year, you had clear resistant individuals and susceptible individuals. In 2020, you still have some resistant individuals and more susceptible individuals. But after about 2020, month 6, you kind of lose those resistant individuals. And you see that change in the phenotypic phenotypes that you see in the field. On the other hand, cercospora is just kind of constant through the whole study. And defoliation, because defoliation is highly correlated with black spot, it had a very similar pattern to what we saw in black spot. So black spot and defoliation, moderately correlated, or pretty highly correlated phenotypically. And for black spot and cercospora, also another slight negative correlation. And we'll see why that's important in a little bit. So the variants explaining the phenotypes that we're seeing in the field, the genotype, the portion attributed to genotype was really low for black spot, and that was because of what we saw earlier in those ridgeline plots. Because there's that shift of the phenotype that we saw, that you had all these environments where you had resistant and susceptible individuals, and then later on you kind of lost that. When we looked at this by year, if we did the same variance component estimates by year, the genotype part comes up a lot more. It's around 30%. To go on and tell more of that story, environment was a lot of that variance that we saw, and that makes sense from that ridgeline plot that we saw earlier. Cercospora was really low for environment because it kept that same kind of the resistant individuals, susceptible individuals. And defoliation is kind of in the middle. The G by U is between 10 to 20%. And here's the QTL result. What you see here are the two populations, bright eyes by micro, stormy weather by bright eyes, and each one of these peaks are QTL peaks. The very top is the black spot is scans. The middle is a cercospora scans, and the bottom is defoliation scans. And what I want to bring attention to is this linkage group 5. How you see there's overlap between the peaks of black spot cercospora defoliation. Over here is overlap between black spot cercospora defoliation, kind of the same story that we saw in the diploids on linkage group 3 earlier. And then we see that cercospora consistently has this QTL on linkage group 1 in both populations. So this is just another view. and it's the view of those same QTL, but highlighted in blue or other places in literature. Here's RDR4 that comes from Bright Eyes, and that makes sense because one of our parents here is Bright Eyes. But what's interesting is that with all this new polyploid software, what we're able to do is we're able to now see the parental homolog effect. So these are effect plots, the average effect plot of having any one of the parental homologs at any given position in an individual. So for instance, all of these are linkage group 5 position 61.23 centimorgans. So if an individual in the mapping population had this Bright Eyes 1 allele homolog, then it is most likely going to be black spot resistant. However, it's going to be cercospora susceptible and defoliation resistant. So what we saw earlier in the previous few slides where you had that overlapping QTL linkage group 5, this is what we're seeing. That was actually a black spot resistance QTL, a cercospora susceptibility QTL, and a defoliation resistant QTL. And this makes sense because black spot causes defoliation, that they're in the same direction. That makes sense. Now, this kind of gives us, as a breeder, a conundrum because now what? What do we do? Like these two are either tightly linked alleles for two different fungi. What do we select for? Because if we select for black spot resistant individuals, it's gonna carry cercospora susceptibility, and vice versa. So what we found is that this right here is cercospora susceptibility. This is if the individuals, so on the y-axis is the cercospora severity. On the x-axis is different groups of individuals. So these are individuals that only carry black spot resistance, but nothing else. So it's also carrying cercospora susceptibility. These are individuals that carry that, remember cercospora 1 QTL that we saw, that was actually a cercospora resistance QTL. So these are individuals that only carried that. Now, if we have individuals that have black spot resistance, and by having black spot resistance, having cercospora susceptibility, if we combine that with the other QTL on 1, if we select individuals that have both, then you have generally lower amounts of cercospora. So what this tells us as breeders is, okay, we can select for black spot resistance on 5. We can select for RDR4. But when we do select for RDR4, we need to make sure that those individuals also carry the cercospora resistance allele from linkage group 1. And a lot of this can be done easier now that we had this other SCRI grant, tools for polyploid, and that allows us to go from genotyping arrays or GBS platforms to doing the genotype calling, to doing the alignments, and to do the linkage mapping or haplotyping, and combining that with the phenotypes to come up with QTL mapping. We're able to do integrated QTL mapping now, so we can have related families using DiQTL and have more statistical power there because your populations are larger. We can do GWAS now properly using polyploid models, and now we can even do genomic prediction in polyploids utilizing autopolyploid models. And this thing called View Poly earlier, I talked about how we visualize some of our results. The next slide just kind of shows how it's useful for a breeder, because we can sit here and talk about genetic studies, we can talk about QTLs, but how does that help a breeder? So a lot of these programs, whenever you run them in R, they have really complex data structures that are really complicated, and unless you have like a dedicated graduate student that's going to sit there and sift through everything and make all the figures, it's really difficult to parse out the results. And for example, on the RRD paper, my very first paper, I had to make all the plots by hand, and it was just thousands of lines of code. Now, so when Chris joined our lab, I was like, hey, like one of the things that you want to do is to make these tools more accessible to a breeder that might not be as keen to coding. So what she did was she took these complex data structures out of R after the analysis, and was able to have a click and point interface where this is a screenshot where you see all the QTL that you're interested in, and you can actually look at one chromosome, you can look at all of them, you can have a drop down of all the traits that you want to look at, and then you can select down here the QTLs that you want, and it will produce plots down here of which way is this QT, is this a resistance factor? Is this a susceptibility factor? And from these, there's another drop-down menu that says, hey, okay, give me every individual that has this resistance homolog, this homolog, this homolog, and make sure not to include this guy. And whenever you do that, it comes out with another output that has all the parental homologs that are included. And this is one of the features that I asked for, is, hey, can you give me a list of the individuals that have that? So she added that. She was like, yeah, real quick. And 10 minutes later, I had this function where I selected the homologs that I wanted from a drop-down menu, and I click go. And in a few seconds, I have all the progeny that carry the QTL that I wanted to use. So that's been really useful. That's come out of the Tools for Polyploid grant. So here's just another view of the QTL that we found on both the diploids and the tetraploids for black spot and Cercospora. In the black are the black spot QTLs, and in the red are the Cercospora QTL, and the blue are black spot related QTL or meta QTL that have been mentioned in other papers. And just to see how that linkage group 3 we talked about in the diploids, how there's that overlap, and linkage group 5, how there's that overlap between the two traits. Those are just interesting things. So I want to acknowledge the team, the big team that we work with at Texas A&M, all the people that are there. We really want to acknowledge University of Tennessee, Dr. Wyndham and Sarah for being such great collaborators and being able to like plant all our stuff, and it's been great collaboration. And I think that's it. Oh, is it again?
What size battery is this?
It's double A. Double A, I think. I have no idea. I can't get the thing out, but they can hear just fine if.
But we can't hear you just fine with that noise.
Or I'll just turn it off.